My name is Bridget Piculell, and I am a PhD candidate in the biology department. And so today I'm going to tell you about my research with loblolly pine, but first I'm going to talk a little bit about dogs. So we're all familiar with the different breeds of dogs and the different attributes or traits that we associate with those different breeds, such as size or maybe the shape of the ears. And a lot of those traits are controlled by genes which are coded for in an individual's DNA. And when we consider that a lot of these traits that we're interested in are quantitative, or they're controlled by multiple genes, and many genes themselves are pleiotropic, or they influence multiple traits, it's not unsurprising that we often see sort of an interconnection or a correlation between traits. So for example, dogs that have been bred for running or chasing have long, thin legs, and they also have narrow heads and long snouts. And geneticists have determined that the reason we see these correlations between those traits is that they're all controlled by a shared group of genes. So while selection was for speed, which requires the long, thin legs, the other two traits sort of got pulled along with them because of that shared group of genes. And what I study are trait correlations in loblolly pine. And loblolly pine is a species of pine that's found throughout the southeastern United States in both natural populations and agriculturally managed plantations. And one set of traits that I'm interested in is the association of loblolly pine with mycorrhizal fungi, which is a type of fungi that grows from the root tips of the plant and extends its hyphae into the soil, where it absorbs nutrients, and it passes those nutrients on to the host plant. And the host plant in turn passes sugars down to the fungi, and so they form this symbiotic relationship. And loblolly pine also associates with another group of fungi in the form of fungal pathogens. And these are fungal pathogens that cause things like pine decline or fusiform rust disease. And these are having a really detrimental effect on loblolly populations across the southeastern United States. So there's a lot of interest in mitigating the effects of these. And what I have found with my research is that there are correlations between the association that loblolly pine makes with these mycorrhizal fungi and whether or not the pines are resistant or susceptible to the fungal pathogens causing either pine decline or fusiform rust disease. And the implications of this is that we can use this system as a way to understand how populations will react to multiple, potentially conflicting selection pressures in the face of changing environmental conditions. And additionally, the Forest Service and paper industries have been selectively breeding pines for different traits such as growth rate or resistance to different pathogens. So before we go too far, we're going to need to see how such directed breeding efforts, like if we're breeding only for resistance to certain fungal pathogens, how is that indirectly affecting other traits such as the association with beneficial fungi indirectly through the shared genes that are controlling them?